All right, so right on the inside of the choroid, you have a layer of cells that actually do the work of transducing light into action potentials, and this layer is called the retina. It starts right here behind the ciliary body, and it sits just inside of the choroid, and it wraps all the way to the back. You'll notice that there's a little tube that exits the eye. This is where the retina connects with the optic nerve, and the optic nerve kind of leaves the eyeball right down here. All right. You'll also notice that the dark sclera here, which is actually white, but in this illustration is dark, it leaves as well. This is actually continuous with the dura mater, the outermost layer of the meninges that covers the brain. All right, so this retina is really important, consists of photoreceptors that have this job of transducing light into action potentials. These photoreceptors come in two different varieties. The first type are called rods. All right, and these rods are much more numerous than the other type of photoreceptor. All right, these guys are 20 times more numerous, and these rods lie within this retina. And what these rods do is that they encode dim light, or they can encode dim light, all right, in grayscale. So they do not encode color. These guys are 20 times more numerous than the other type of photoreceptor, and they're really great at encoding dim light in grayscale. All right. The other type of photoreceptors are called cones. These are much less numerous than rods, but these guys can encode color. In fact, we have three different types of cones. We have cones that encode red light, blue light, and green light. So just like your computer, RGB. And when color stimulate multiple types of cones at the same time, so if you have multiple types of cones that are stimulated at the same time, that will allow you to see obviously colors other than red, green, and blue. Okay, it lets us see the whole color spectrum. All right, so by kind of combining which type of cone you stimulate. The only problem with cones is that they just require a lot more light to be stimulated, and so they don't do nearly as well in dim light. And this makes sense. If the lights are very dim, you'll notice that it's nearly impossible to see colors, but you can still make out the shapes of the objects that might be around you. All right. How these guys are kind of set up is quite interesting. So if I were to just zoom in on a little part of the retina right here and kind of redraw it down here, what you would see is you would have, I'll redraw this. This is the back of the choroid, okay? And inside this retina, you'll find that these little rods are going to kind of look like that. You'll have your nuclei right there, and they'll have these little like oval shaped cells that lie right beside the choroid. There's a very thin layer of the retina. It's called the pigmented layer of the retina. It lies right behind these rod cells, and the pigmented layer of the retina, which is right there, we'll just call that pigmented. Can you see that? Yeah, pigmented layer. That just contains basically some macrophages that work really well in the renewal process of all of these different cells. They help kind of renew these cells when necessary. So you have all these rods that kind of lie right there. You'll have much more rods than you do cones. I'll draw a cone in black. All right. You'll have these rods and cones. And what they are all connected to, they're actually all connected to a bipolar neuron right here that lies right beside all of these cells. And then that bipolar neuron is attached to an afferent nerve or neuron that leaves the retina in this direction and then leaves the eye. So if we were to follow these afferent nerves, it would start here, it'd come here, and they would exit out the eye right here in this little tube. Now this little tube is called the blind spot. So right there is called the blind spot. And that's called the blind spot because there's no photoreceptors in this little area. And you literally can't encode any information in that region of the eye. And you don't notice this because your brain does a really great job of kind of filling in what it thinks should be there. And so you don't really notice that there's a blind spot in the eye. All right. Now, one more thing about the retina here is that there's this location right at the back of the eye, and we'll draw it like right here, and it's like a little bulge. It's like a little bulge in the retina that's right behind the cornea. This guy is called the fovea centralis, and this fovea centralis contains a very high density of rods, and this is actually the only part of your visual field that's in focus. Most of our visual field is not in focus when we're looking around. If the only really part of your vision that's in focus is